All right, let's get started. Hi, everyone. I'm Bree Price. I'm the WSU Bee Program Extension Coordinator. Thank you so much for coming to another webinar in our 2025 webinar series. I just have a few statements to say before our speaker gives their presentation. So the WSU Honeybees and Pollinators Program is a cornerstone of the College of Agricultural, Human, and Natural Resource Sciences, abbreviated CONORS, that is dedicated to fostering resilient ecosystems in Washington and beyond. Our mission intertwines innovative research, community engagement, and education to safeguard pollinators pivotal to our food security and environmental health. In partnership with the Connors Resilient Washington Initiative, we're committed to advancing sustainable practices and pollinator-friendly landscapes and ensuring a flourishing future for agriculture and natural resources. We have 5 more webinars in this webinar series this year. For more information about those, please visit our upcoming events page on our website, bees.wsu.edu. There will be time to answer questions today after the presentation. Feel free to type your questions in the Q&A box below any time during the presentation. After the event and before you close your browser, you'll be prompted for a short 5-question education and outreach survey. Your responses to this help us understand our impact and make our education and outreach more tailored to what you're wanting to learn about. Today's speaker is Dr. Dewey Karen. Dr. Karen is an emeritus professor of entomology and wildlife ecology at University of Delaware and an affiliate professor at Oregon State University, as well as a content communications specialist for the Oregon Master Beekeeper Program. Since university retirement, he remains active in bee education with meeting presentations and writing. He's also a past president of the WAS in 2010 and currently is a member at large to the WAS board, as well as a coordinator of the WAS mini conferences. He is an author and contributor to the Honeybee Health Coalition and USDA Pollination Protection BMD web program. So he is very well known in this field. This webinar is titled Deadout Necropsies. How did your colonies do last winter? Dead colonies have a tale to tell. This webinar will teach you how to look at deadouts to determine why they didn't survive and came through winter very weak, and some things you can do to help ensure better success next time. Okay, Dewey, I'm going to mute myself and hide my video.
Okay. Thank you for that introduction, Bree, and for getting this webinar series together. I've enjoyed it, the gardening and the nutrition ones that have already been presented. As indicated, we're going to talk about our bees. Maybe some of you had a chance to be in them today. And I hope, you can celebrate with your bees like the cartoon here is we made it. Some of you I know have not had a chance to look at your bees, other than maybe seeing if there's been a little bit of flight, some pollen coming in. But the weather is going to improve, and spring is here now, and so hopefully the bees too. If your questions don't get answered, or you like to have a further dialogue, I have my email there. I have both University of Delaware, so that's dmkaren@udel.edu, or get in touch with Bree, and she'll send the questions on to me as well. All right, I do hope your colonies have made it, but as indicated, the dead hives do have a tale to tell. What we're going to talk about then today is looking at those colonies that don't make it. All right. Let's see. I'm really pleased to understand that the program at WSU has a brand new person to see that studies, joining a great a good crew that's there already. As we know, Dr. Priya Basu, she just joined us first of the year. Priya was with us here at Oregon State on a postdoctoral position after she finished her degree in India. and you would really have a real winner in Priya. Very, very strong program. So congratulations to Priya and to WSU for a great decision on someone to help replace the retiring Steve Shepard. Big shoes to fill, of course. You've probably seen the headlines. Survey reveals over 1.1 million bee colonies lost, raising alarm for pollination and agriculture. That 1.1 million, that's in a context that we think we've only got about 3.56 or so colonies in the United States. So that is a huge number. The survey was done by Project APIS-M with the concurrence of the Honeybee Health Coalition, which I'm a member of, American Bee Federation as well, and American honey producers. And they surveyed 68% of the owners of bee colonies, the estimated number of bee colonies in the United States. So very, very robust to come up with this number. And the average loss of the commercial beekeepers was just an incredibly huge amount, over 50%. And backyard beekeepers, those with less than 50 colonies, and what we call sideliners, those that have other jobs but have a number of bees anywhere between 50 and 500, also losing over 50%. And this actually is the second year of this very heavy levels of losses of bee colonies. I conduct here in the Pacific Northwest. This will be the 16th year now of a PNW survey. And the survey has several components to it. The component that looks at losses is asking for the number of colonies you had in the fall, and then how many in the spring. And the way to get to this, it's an electronic survey. Way to get to it is just simply PNW Honeybee Survey, all one word.com. And then you'll see a top bar. And if you click on Take the Survey is now open. It'll go through the month of April. And as I indicated, it's not for brand new beekeepers, but for those that have had colonies that did overwinter some colonies in the fall. I'm not looking for those necessarily that had a very poor success or those that had great success. I want both groups of those that will take and enter the data. Should take you less than 5 minutes. There's a second component to the survey. The second component is management, things you did for wintering, things you did for feeding, things you did for mite control, etc. But you do not have to answer that, particularly if you've got several colonies. It'll take a while, a little bit longer to do that. So you can fast track, and so basically I'll get your information on the losses. And for those clubs throughout our area, through the Washington, the Oregon, Idaho, and then the California, the clubs where I get more than 20 member responses, I give the club an individual report of what the members of that particular club had. And because I have been doing this for 15 years, I have a record of how this year looked in comparison to other years. And what I would hope you might do with this information is after you've had a chance to see how you are doing, it's a chance to, in your club, to see how you are doing relative to your club, but also relative to your other beekeeping neighbors in other clubs and in the state, to kind of have a reading in terms of how the winter went and how you were doing with your particular management. PNWHoneybeeSurvey.com. I hope if you haven't had a chance to do the survey, you will join and give me some information this year. And I appreciate all those that have and those that will give some information. It's open through till April. So, deadout, something that we're faced with. Maybe you've already been faced with it, reading deadouts. This case at Club Apiary here in the Portland area in March, we saw the queen. The colony was weak, so we fed dry sugar, and you can see a piece of paper there. We fed dry granulated sugar. But when we got back in April, the colony was dead. And so in my hands here, I'm looking at the dead cluster. You can see I've got two frames, and there are some bees down below. So this was a starvation colony in the spring. We tried to rescue it with the feeding of the dry sugar in March, but it was not enough. They've run out of honey. So the frames I'm looking in, there's not a drop, not a bit of honey left in any of the cells. And even though there was still a little bit dry sugar left there, the dry sugar just was not enough with continuing cool weather to get them through. The reason we did not go with a liquid sugar or a different method of feeding, such as fondant, or whatever is just by preference. We thought March was too early for a liquid sugar, and we had the granulated sugar available. We did not have the fondant sugar. But there was enough of a cluster of bees there. The heat is released by the bees to keep warm. That heat rises, and then it would be make a slurry out of that granulated sugar, because bees can't really eat the sugar itself with their mouth parts. But it turned out wasn't enough. It was not enough bees to try to keep the colony warm, and certainly not enough honey stores. So why did the colony die? Well, sometimes we don't know, or we can't definitively say it was for this reason or that. But we then need to look at the clues. So dead hives have a tale to tell. So let's do a little bit about reading deadouts. Now, I do have this chart. If you'd like to have an individual chart, I'd be glad to send it to you. I'm not going to cover everything in there, but I'm going to try to emphasize that in spring, we can lose colonies to things such as starvation, freezing. Sometimes the cluster splits, and the two splits of bees are just not a critical enough mass, and so they freeze, or aspects of too much moisture. However, we are going to emphasize the most common major factor in loss, and that is due to mites. What we might call a number of different things: BPMS, B parasitic mite syndrome. Some will call that parasitic mite brood syndrome, because that's one of the ways that we will read this. Then, of course, there are, and I'm going to come up with some examples here in just a minute or so. But the last group is there are some other factors that may be the reason why your colony did not survive, with predation by small mammals, larger mammals, small hive beetle, the fact that we're going to see scavengers, and then a series of other things that, quote, can go wrong, such as absconding, loss of a queen, exposure of the colony that gets tipped over, or the covers blow off, etc. So there can be some other factors. So we're going to go through each of these as trying to then retell from our dead hive. It's not going to bring them back, obviously, but maybe it could be a factor to help us avoid a subsequent loss in another season. Okay, so with that in mind, let's go and take a look. To read bees under stress, bees that are not healthy, we need to start with bees that are healthy. So what we want to do is in the fall, as we put them down for our last inspection, is make sure they have some honey. We can see in the frame here in the top left and top right, some capped honey. We want to see some brood. In the fall, this brood are going to be bees that are not going to do the normal work of colonies. They're not going to be nursing much more brood. They're not going to be processing much more nectar coming in. They're not going to be building comb normally, and certainly not doing much foraging. And all those things help them preserve their vitellogenin. And that then also affects the hormones that key how long they're going to live, but also they're going through their sequence of adult activities. So these bees that are in the fall are the ones that are going to have to live that period of time, and they're doing it because they're not starting in immediately as bees that are cleaning, and bees that are nursing, and bees that are processing food. Saving doing all of those, then they have the food resources in their body, and hopefully in the hive as well, to get through the winter. So healthy brood looks somewhat like this pattern. The middle portion of all those open cells is the brood that is just hatching. So that's normal because those are the bees that are coming out, the brand new adults. The brood, you can see the cap brood is the easiest of the brood stages to see. We're looking for the pattern. We're looking for consistency of the pattern. Relatively few cells that are open that do have a developing pupa. Those that don't are the open cells here, these black cells that we're looking at in the area of the cap brood. Okay. Now, if we look during the active season now in spring, where those bees are emerging in that pattern in the middle, the queen may already come back or has come back. And if you look closely, you might see open brood being the eggs or the larva. But it's much easier to look at the cap brood to get that reading. Okay. So that middle part isn't a mystery if you recognize that some of those cells around there are just coming out. The bee, you can see her waving antenna and maybe her feet. The individual bee that's in her cell has to get herself out of the cell. The workers on the outside are not helping her do that. But the workers on the outside are the ones that put those wax cappings over the cell. So if we're at all in doubt, we can sacrifice some of those developing bees. So here we've taken a developing larva out of the cell with a toothpick. We want to see that it's white and that it glistens. It's not an off color, yellow, certainly, or brown. Those signify unhealthy conditions, some disease conditions, starvation, which sometimes will occur in early spring or late fall because there's just not enough bees to keep their whole brood rearing area warm enough that when you particularly have a new queen that's capable of laying lots of eggs. So if you're in doubt, check it out. Check out what's healthy. Here's a look at some of the open brood. The eggs are the 5 cells to the left side, and then there are 6 cells that are evident that have very young larvae. We call the larvae instars, and because bees have an exoskeleton, they have to reabsorb their body covering, their exoskeleton. They don't have bones, it's all in the covering on the outside. And then they put down a new one. So these are very new larvae, and they rest on a pool of food that is provided as the eggs are about to emerge or change into the larva. The eggs lean over. So these 5 eggs are very evident because they're not standing straight up looking at us, but they are leaning over, signifying that the embryonic change is going on, and soon that egg will then become that instar larva. Now, one thing we might look at here is that what we want to see with the feeding stage, that's the larval stage, that there's a generous supply of food. In this case, there is not. Where that question mark is just above there, that larva has a very small amount of its food. They literally rely on a pool of their food. And it's also food that is clear. What it needs to be initially is a cloudy mixture because it comes from the glands of the bees, the adult bees that we call nurse bees. So this is a indication. We see this in spring. We'll sometimes see it in the fall. And this is an indication that there is stress. There is some stress. Perhaps there's not enough new nurse bees. Perhaps the nurse bees are all older bees, like in early spring, and they're not as efficient at their glands operating and producing the brood food. And so the larva still can hatch into perfectly healthy adults. Sometimes they'll be a little bit smaller bodied, and because of lack of some of the foods, they will go through some of their life stages a little bit faster than would be the normal case. So looking at what's healthy, the brood here looks very healthy, except we would say that maybe we'd like to see a little bit more brood food in those open cells with the larva. What we don't want to see, of course, is the spring deadouts. Looking at 2 different ones here. On the left, that's a half depth body, medium depth box on top of a full depth. The bees have moved upwards you can see where the red circle is, the queen was laying some eggs because those are cap brood. These are the individual cap cells. You can see the cluster of bees now dead when we discovered this, on at least at least three frames in the top box. And if we then went to the bottom board, we'd likely see a number of other dead bees that have accumulated on the bottom board that the workers have not had a chance to clean. They're up here in the second box, and they're not down on the bottom box except those rare winter days when they can get out and get some pollen. But they are dead. So this would signify that we had a queen. They had started their brood rearing in very early spring, but then for whatever reason, like that very first illustration, maybe alive in March, but now dead in April. The illustration to the right is a full depth, and there we can see the dead bees clustered. This is what they will do. That's their behavior to help survive over the winter. But they are dead at the time that we discover them. They are on the area where they did a rear brood. But in both of these boxes, we don't see honey, or we don't see honey immediately, at least where the bees are. So we would think that this combination of factors, bees starting to rear their brood in spring, bees that cover at least three frames, more perhaps on the full depth, bees dead on the bottom board, but where the bees are dead immediately in that area, no honey, no capped honey, nothing available for the bees to feed on when we got that later season cold snap. And I guess then the diagnosis, our reason we think the colonies in this case didn't survive would have been starvation. So here's another example. We'll dig into this one a little bit more. And so I've asked a question looking at this. What do you see? What is going on here? At the arrow, the blue arrow that I brought in, we're looking at a bee bread, stores of bee bread with a honey glaze over some of the cells, not some of the others. Probably some pollen that's come in. Now, this is also a dead cluster. Look up here on the top bar, looking at what we see there is we see cells that are not fully formed. We see cells that are not six-sided. We see some that are five, some that are seven-sided, and we see some that are filled in. This is propolis. This is the bee resins that the bees have collected, and they're reinforcing the comb. So they've taken the beeswax and mixed it with the propolis, the bee resin mixture, and that just helps make for a stronger frame. Some bees, such as the bumblebee, such as the stingless bees of the American tropics, they put the two together all the time. Our bees kind of keep them separate. So most of the cells are just comb, beeswax, and then here at the top, beeswax with some propolis. But looking at the dead cluster, doing our diagnosis here, what do we see? Some of the bees have been removed so that we can see a little bit better. We will see overall on the colony that there are dead bees often there on the bottom board. There's no honey on this particular frame, but there might be on some of the others. Our beekeeper in this case did not do any mite treatment or waited until very late in the season, September, October, to try to do some mite control. Generally did not know if they had mites or the number of mites they had, so had not been doing any mite sampling, or at most did one or two late in the season. If you look in some cells, you might see mite poop, which is the guano, the remains from the mites. They will mark the beeswax cell, and it will show up sort of as a white blob in some of the cells. You might see some cap brood, but we can't see any in this particular photo. Now, a couple of other things. Let me zoom in on where that circle is, and ask you again, what do you see here? All these bees are the bees that were in that cluster. We're trying to stay warm. So the cluster has a very tight outer circle of bees, outer perimeter of bees, very tightly grouped together. And in the middle, it was fairly wide open, because that's where the queen generally would be. Those are the bees that would be producing more heat with more movement. The bees on the outer side being really quiescent, just trying to retain the body heat that they have. And as the bees on the very exterior get colder, they move towards the interior of the cluster. Very cold nights, this is a very tight cluster. Very warmer nights that only get down, get down into the 40s or so. It's a looser configuration. But looking closer, we can also see that here in nearly each of the cells of this middle part, there are dead bees right in the cells. So they're head first, and we're looking at bee butts. So every cell, virtually every cell there, has a bee in it. And this is a measure to when there is no brood to try to keep warm, keep the bees more compact. So this would signify that there was a period when it got pretty darn cold because bees crawled into all of the cells as a way to get more compact, to make the cluster a lot more compact. We can also see the queen. So there was a queen in the colony. We don't see brood where the bees are here. Perhaps there was some brood, perhaps some brood that is already capped. In this case, no brood because virtually all the cells have bees inside. Now, when she's laying brood in the winter, she will not be very trying to put an egg in every cell because heater bees can move into cells that are empty, really elevate their body temperature and help keep brood in all the cells around the cell that she's crawled into. But in this case, no brood, no cap brood that we can see. So again, we need to try to make that diagnosis. Now, in this case, because it looks like this is somewhat fresh pollen, it doesn't have a real good glaze over the cells. Maybe they made it into spring. And if we've got all these characteristics, particularly then the dead bees on the bottom board, a colony that all of a sudden there was a very deep cold snap. They tried to keep warm enough, but they didn't have enough honey to try to keep everybody warm. The cold snap that lasted a couple, 3 days, and we had a spring starvation. Okay, so we don't see honey in any of the cells. The bee bread does not have much in the way of carbohydrates to help them run their wing muscles. So the spring starvation, often seeing this big pile up of dead bees on the bottom board. And the other clue that we need to look for is, did we have that sudden snap, that sudden cold snap? And maybe you saw bees flying and bringing in that pollen before the cold snap, and then they just couldn't get through that last of the winter cold snaps over the winter. So in instances of spring starvation. We can get starvation of the colony in the fall, highly unusual because we generally have fall sources for the bees, and if there's not, we know we need to do some feeding. So usually starvation is a spring event, very frequently after they have started rearing some brood, and then we got a cold snap. So look at these clues in terms of trying to make that diagnosis. And really, it is not as common as we would believe. Um, what is more common is this situation. There's our frame. In the corners, there is some honey. The dark area is where they had brood through the year, and a pretty good brood area. But you can see there's not enough bees to cover the brood. And in the brood area, there are cells that are capped over. And if you take a forceps or a toothpick and push off that capping, you'll see there's a developing bee inside. perfectly normal looking developing bee. A bee that should have hatched, come out from the cell as an adult and joined the population in the fall, but didn't. It didn't because looking at the size of that cluster. Now, the bees here are thoroughly covered with a mold that's growing on them. But comparison to the last slides I showed you on starvation, notice how few bees there are. This is and a corresponding on the other side, or the corresponding side of the frame that I've already removed. This is the extent of that cluster. So it's a small population. They have honey stores, but they're not immediately on the honey stores. The honey stores are over a couple inches from where they are. We see the moldy bee bread. Those are the bright white cells. And that means that the bees were not there where they should have been, over that brood area, keeping the brood warm enough so those bees joined the overwintering population and kept the mold from growing on top of their bee bread stores. There were not enough bees. So this is an instance of what we see in terms of what we call freezing. What happened, it was not that critical mass, the necessary number of bees to get them through winter. So too small. Sometimes this is a split, particularly we see that in hives like longitudinal hives, top bar hives, sometimes in our wary hives, because the honey is split in two directions. And of course, this cluster, as it forms, as it gets cold enough, it can only really move one direction to keep in touch with the honey store. So longitudinal hives, top bar hives particularly, the honey is stored on either side of where the brood area is. The brood moves to one area, some of it moves to the other area, and you get two small groups of adult bees that cluster, but there just is not enough bees to keep everyone warm and keep honey circulating through that, and particularly also keeping their bee bread stores and the developing adult bees from coming out from cells and joining their population. So too small. They just got too cold. Not enough honey to keep them going. Now, sometimes we'll see a situation that looks like this. So here's that brood below. They're still on brood, but they've abandoning brood below. So the population is decreasing. And then on the top of the cluster itself, we see moldy bees. Not so much the mold of the cells, but on the bees. And this signifies, many cases, that there is moisture that has been our critical factor of the bees not being able to survive the winter period. The bees are living. They're not dead. They're not in a dormant stage, although we'll call the winter clustering dormant. And so they produce heat, and heat rises. And if that heat rises to the top of the hive, and the top of the hive is not insulated, then that water can form as droplets. If the top is insulated, then the warm air will be below a dew point and will then spread out to the sides and then fall back downwards towards the bottom board along the side of the colony. But if that top is not insulated, that moisture can accumulate on the inner covers of the colony, droplets of water form and then fall back down on the bee. So here's the cluster, and we can see on top of the bees where water has dropped down onto the bees, and that's where we're getting this moldy configuration. Non-insulated, non-top insulated colonies have a tough time in our climate getting through the winter. And our enemy in this case often is that moisture, because as the bees get wet, they get more active. That means they have to eat more food, more honey to try to dry themselves out. If you do any winter sports, I'm sure you're aware that if you keep your body dry, it doesn't matter necessarily how cold it is or how much snow there is, but once you get your clothing wet, then you get cold quickly. Well, same too with the bees. Once they get wet, this water dripping down on them, they get much more active, trying to dry out, consuming much more in a way of stores. They then get clustered together, abandoning the brood that you can see here, the cap brood, and the moisture becomes our real issue. That is solved with better top insulation. Let that moisture then spread out, go to the sides of the colony where it is cooler, and let it condense at the sides of the colony, falling down to the bottom board, but not falling down onto the bees in their cluster. So too much moisture. We'll also see the bottom board is just soggy, wet, very wet, wet with the bees that are dead on the bottom board. And again, this is not a very big cluster of bees left over, in part because they are having to work to try to dry themselves out. And so they're using up their body reserves, and not living as long, and we get the small colonies again. So those are some factors that we might see. We see too few bees, a little bit more common than we see the starvation. However, number one on our dance card here is mites, varroa mites specifically, the condition we can call as PMS, parasitic mite syndrome, or parasitic mite brood syndrome. What we will see is the characteristics now is like the colony is there, but nobody home, or very few bees home. We're not seeing any bees here on this frame that we pulled out. Seeing relatively few bees that have fallen to the bottom board. They're just not there. And why they're not there is the bees that are raised in the fall, if they've been fed on by the varroa mites, that's how the varroa mite reproduces, feeding on the bee brood, the pupal stage. If they've been fed on by varroa mites, they leave home. They don't have the necessary resources. They are sick, they are stressed, and they will be leaving home. And so by the time we see our colony in the spring, no one is there, very few bees. But what we see in the brood area is the remains of brood. So all these cap cells you can see here are the remains of brood. There's a little bit of honey in the corners of this frame, not very much, but it's enough to sustain some bees. But what has happened is the bees have left the brood because the population kept decreasing. Now, some of these colonies will appear as dead colonies in the fall, or now as we open them up in the spring, we'll see that they're not living. So here's our brood area, and you can see cap cells. Now, a number of the cap cells are perforated. There's holes in them. Some of the bees are trying to get out. You can see the heads and front legs out, but that's as far as they were able to get out. It was just too cold. And the very bottom of this frame, you can see some capped, bee bread stores, normal bee bread stores. And as I indicated, there is some honey on this frame. That's where on the right side, some honey up there. Those are the capped honey cells. Here, give an example. Here's a lot of honey. You can see maybe the bees were on at one time because in their cluster because of the darkening of the cappings from their feet, and also the cappings sink in as they cluster on it, and then they get this watery appearance. So off to the left, the normal looking at cappings, and then the watery cappings. But look at that brood area, a huge brood area. Look at all the brood cells that did not come out as adults. These are all bees that needed to be in that colony as adults to get through the winter, but they did not. And it was because of being fed on by bees. So whether we see it in the fall, we see it in the spring, this parasitic mite syndrome. So here's a lot of honey, still a lot of honey left in the colony, but not enough bees born into the population in the fall to have them get through the winter. We see it in the spring or summer. Now, in the fall, we can sometimes diagnose this, but it's pretty subtle. Okay, so here's a frame from the fall colony. So this would be a September, maybe October colony. It's a medium depth. You can see very solid brood pattern here to the right side of the brood pattern. Nice looking brood pattern here. But as we go to the center, it looks a lot more spotty. Now, some of this may be due to bees emerging, but it's also an indication that maybe, just maybe, the bees are finding mites in the cells and they're removing the mites. They're opening the cappings of the cells. The founder's mite runs out, but her reproduction does not continue. If they find the mites, they take out their developing pupa. And even if they don't find the mites, other bees come along. Whoops, this cell should not be open, and they cap it over. So, what are we seeing? Well, we don't see enough adults in this illustration. Okay, the population already in August, September is this was a September photo, are low in adults. We see that spottiness of the pattern, but starting out is solid on the right, the latest eggs that she put in the cells, but spotty towards the center. And then we can actually see brood that doesn't look right. We call that snot brood. And it's not a European foul brood type looking dying larva or an American foul brood dying larva. It looks like the developing larva is not normal. It's not that C-shaped, nice tight C-shaped and glistening and white structure. It's this decaying looking, graying, just looks like what its name it signifies looks like sorry for this, a piece of snot in a cell. Okay. Then we'll see some mite poop. If we look in the cells there in the center, we're most likely to do so. And we'll see cells that the bees are actively uncapping. This is another frame. This is what we're calling this spotty brood pattern. Instead of a solid pattern of cap brood is just all over the place. Now, the queen, likely laid eggs in most or nearly all of the cells, but the bees were taking and removing them because cells had mites in them. You can see some white in the center, and those are larvae. Lots of the cells have larvae. It looks pretty solid, but again, once they reach the cap stage, then that's when the mites will be feeding, and the bees will find and remove them. So here's a close-up of what that looks like. This is an early stage of bee parasitic mite brood syndrome. We don't know what is affecting it. This is a viral infections. We know some. Usually it's one of the patterns of deformed wing virus or Israeli parasitic virus. But this is what it looks like. IBDS is idiosyncratic brood disease syndrome. And that's a fancy name to say we don't know why they're dying, but they certainly are. So we see lots and lots of uncapped cells, okay? Spotty brood pattern, not that solid cap cells. We're looking at what we call hygienic bees. Those are the adult worker bees that are walking along and they detect in the cell a mite reproducing. So they remove the cell capping. So the white we see are these removed cappings or cappings in the process of being removed. So in this circle, there's, let's see, 1, 4, 6, 7, 8. 8 of those roughly 25 cells are now being uncapped. And because there may be mites in some of them, they will then yank out their developing potential sister, yank out the pupae. And so what's happening is we're not getting normal birth into the adult population of the colony, the bees that are going to have to live longer in the winter. So this is a severe mite condition. And the bees are doing their job, trying to get a handle on getting rid of those mites. But in so doing, there's not going to be a big enough adult population. So hygienic bees doing what they should, but unfortunately, we don't have necessarily the population we need. What we will see, let me go back to the second circle I have there, we will see in some cells recapping. So I have 6 cells here The top three look like they have not been uncapped, but the bottom three are not as smooth, not as slightly convex, I'm sorry, popped out just a little bit. Not as much as drone cells, but these are worker cells. But you can see sort of like a dimple in the middle of some of them, a little sunken area. These are cells that have been recapped. There wasn't a mite in them, and other bees, not the uncapping bees, not the hygienic bees, but other bees have come along and salvaged their developing pupa. So those bees have a better chance of joining the adult population. Unfortunately, our breeders are working on the hygienic properties, that is the finding and uncapping, and not on the recapping. And so that if we have high numbers of mites, a lot of cells are uncapped, a lot of pupae are removed, and we don't have the adult population going into winter. Now, I did mention snot brood, and so here on the very bottom board, this is a plastic frame. So this is the plastic bottom there, that line you see, the white line. On either side of the cell cap there is this snot brood. So you can actually look into the cells, and it's not a normal developing larva, not at all. It's not white. It doesn't glisten. It's sort of lying on the bottom of the cell. It's a decaying, a dying larva. So that's what we mean by snot brood. Okay. And we have brushed the bees away to get this illustration, but we would also see that low adult bee coverage, and particularly often when we're in the colony, they move off brood. And even though it might be a cool day in September, October, the bees are moving off the brood, and they're going to, as they get fewer and fewer bees, they will continue to move off the brood. You'll see cannibalized brood. At the lower, I have 4 yellow arrows. And what they have found there is the cell had a mite in it. The adult founder's mite ran off, but the bees are now getting rid of the pupa. So these are in various states of the bees getting rid of them. So they actually chew them down. And these are cells particularly that have mites, but also with a hygienic trait. And the more hygienic traits you have in your bees, the more they're going to break down those cells and get rid of the remains. And again, these are bees that should have joined the adult population, but will not have a chance to do so because the bees are cannibalizing it. So what we're doing and doing the looking is, of course, the bees do all the work. We are the quality control. Okay. Even though there are disease inspector bees, bees that spell the mites inside, bees that uncap the cells, those hygienic bees, et cetera, et cetera, we still have to do the quality control. This is a book by Albert Chuak, Utah beekeeper. Do You Speak Bee? And he has these beautiful illustrations of different jobs that bees do. So here's the disease inspector bee that he has. He's talking about what we will say, oh, that's what a bee is doing, an undertaker bee or a guard bee, a nursing bee, etc. Now, we've been talking about mites. I've been talking about mites, but it's not the mites. It's actually what is really going on is the mites are transmitting or facilitating the transmission of the viruses, particularly 2 groups, the DWV virus group. That means deformed wing virus. Those bees in the circle, all of them have wings that are not functional. These bees will not live the winter. They will not obviously become foragers. If the bees are on cells that are recapped, the mites already have fed on them. Some of these come out with the expression of the virus without wings. And of course, these are not going to be bees that are going to be very helpful. The other group of viruses are the bee paralysis virus. There's a chronic CBPV, chronic bee paralysis virus. There's an acute that kills bees much more rapidly. We do know with colony collapse in the spring is really acute paralysis virus. So there are a bunch of the paralysis viruses. Seems mostly that it's the deformed wing viruses. Our population of deformed wing has at least 4 variants. and what we call DWVA is being replaced by DWVB, and that seems to be more virulent. And maybe that's the reason we've had such terrible, terrible losses among our commercial beekeepers over the winter. So a recap on the mites, few adults, no really dead adults in the colony. You'll find this kind of situation scattered, leftover, sunken, perforated, capped brood. You'll see the larvae are being chewed down in the fall. You'll see the remains of some of that in the spring. Adults not able to come out of their cells, partially out of their cells, their mouth parts and their legs, front legs sticking out, but wasn't warm enough that they could come out because their brood was being not being properly kept warm with the adult cluster of bees over top of it. Dead bee removed, and then this grayish snot brood, not yellow, not that ropey, goopy stuff that's American foul brood or yellowish that's European foul brood, but a grayish brood that we call that snot brood. Recap. All right. So there are some other factors. Let me finish up then in the last couple of minutes I have here. Certainly we can see things inside of the colony, mice nest, shrew evidence. That's a disturbance. They don't eat a lot of bees, but it's continually disturbing the colony. Mice, particularly, will construct a nest, taking out comb. They'll pee on other combs to mark them. Just a mess. And of course, the bees stay away from it. And so the whole nest you think is for the whole hive for the bees is not. There's something else in there. A larger mammal predation, primarily skunks, but also we think raccoons, large animals that might tip over colonies. And of course, for a number of us these days, colonies that are just scattered the equipment scattered all over the place, emblematic of bear damage. And those colonies that are opened up, the colonies that are continually disturbed by the skunk feeding on as many as 100 or more bees. It scratches on the outside, bees come out, it gobbles them up, chews them, and then spits out the cadavers. But that night after night disturbance, you can really reduce the population. Small hive beetle, we have adults in our area. The insert there in the middle shows that adult, but in areas where they are able to reproduce their whole life cycle, the small hive beetle larva will make this slime material, and they really take over part of the colony, like the mouse nest is doing, taking over part of the colony. And then the bees avoid it, and those are reasons in some colonies where you'll get the bees to actually leave because their home is no longer very attractive due to small hive beetle, due to the mammals in there, mice, for example. And then it's the behavior in the fall that we call absconding. And then the scavengers, if we have our mild winter like we had in December, most of our yellow jacket nests are gone, but not all of them. And so the bees that are trying to get through the winter, trying to bring in a little bit of pollen in December or those nice days in January, are picked off by the scavengers, and particularly yellow jacket. Inside the colony, as the bee population decreases, then wax moth eggs that are there, the wax moth adults enter at night, lay their eggs, then they damage the colony. And again, this external predation of these type of scavengers can be a factor. I mentioned absconding. We don't know all the reasons why our bees abscond. It's very common behavior in Africanized honeybees, not as common in our European race bees, but it does happen. These are the bees that simply just get up and leave in September, October from our colonies. The absconding, the bees that leave, really don't have a chance to survive and get through the winter. And of course, all we find is a colony that has everything in it, but no adult bee coverages. Pesticides, certainly the level of pesticides at some of our bees, if you are all in an agricultural area, number of pesticides, and it's sort of that pesticide soup that might be a factor. Again, stress on the bees, not able to do as well in terms of the vitellogenin put into the fat body, the glycogen, which is their carbohydrate storage in their bodies. Pesticides affect all of that, of course, and then actually can affect the viability of queens in their being born into our colonies during the year, and in the mating process with males, the drones being affected by levels of pesticides, not able to effectively inseminate queens. So all a number of bad downstream things happen with pesticides. We certainly have queen loss. Queen loss at an inopportune time means the colony doesn't get big enough, strong enough, store enough honey, or if this occurs late in the season, very early in the season, we don't get the proper matings, not having a large enough drone pool. In the fall, particularly, the drones, our older drones, may not be able to provide enough live sperm. And so, although a queen can mate in September, if she's born into a colony, often we don't get the proper matings, or in some cases, not mating at all, depending on the fall weather. And there may be a queen, but she's not there. And so that'll come out as a colony that really has only drone brood in the spring, or a colony that just is not getting started with its brood rearing like the rest of the colonies should. And then the accidents, the blown off covers from the large animals, for example, the bears, possums spreading stuff out on us, or raccoons, the covers that fly off in the winter, the soft earth over the winter with all the rains and the colony falls over. They may survive if we catch them in time or get that cover back on time, but it's obviously levels of stress that is not good for the colony, and so other factors as well. So number one, the mites, certainly in the spring looking for starvation, too small number of bees, or perhaps one of these other factors is the tale that our colony might tell about us. But it's mostly all about the mites, parasitic mite syndrome. Spraying mite control. I want to say one final thing on mite control. We think in our bees, that's the blue line here, roughly a bell-shaped population. When they get to be near the population peak, we have to do swarm control. Early in the season now, we're not as concerned about it. Well, our mite control has always looked at this intersection, this August, September time later in the year. To be effective in mite control, we need to keep the populations low. So spring, now, this month, last of March, certainly April, we need to get on our bees and do our mite control this time of year. We can't bring high mite numbers down in the fall. We don't have the proper tools, or we have to repeatedly use the tools, which is annoying and time consuming, and really stresses bees out with all of the use of our pesticides. In the spring, we don't have to use pesticides, or we can use a pesticide treatment that will be mighty effective, and we don't have as high a number to have to deal with in the fall. So spring mite control starts now. Here's a toolkit for your diagnosis: gloves, forceps, toothpick, or a nail, capping scratcher. That's a way to look into drones, remove drones. Our method of taking a sample, powdered sugar or better, alcohol. Our kits, we have kits that you can, if in doubt, that you can then take a sample and determine whether it's a disease such as one of the foul broods, the bacterial diseases. Certainly flashlight, magnifying glass, record book, and something to put your containers in. So good time now to think about your toolkit, what you're going to do if you can do that diagnosis during the year of what might be some stress and how the colonies are doing. Let's go here. What we're doing here, and Bree and I are working on a flyer that will give you some more information Honeybee colonies that failed over winter may have a tale to tell. So we're working in terms of a flyer. The chart that I showed you had some more information if you'd like to get a copy of your own. I can simply attach it as I send it to you. So my email is dmcaron@udel.edu. And a couple of resources in my book, The Honeybee Biology and Beekeeping. You can order that directly from me, from Wickwash Press, the publisher, or pay more money and get it from Amazon if you like. I'll be selling them at the fall WASPA meeting. I'll be at the meeting this fall. I'll be at a number of different clubs coming up. I'll be at Clark coming up. I'll be at Olympia. I'll be at Lewis County coming up next month, the 9th, I think, next month. And also for that, everything you need to know, possibly can use for Varroa is this Tools for Varroa Management from the Honeybee Health Coalition. We're working on a new edition. It doesn't have the oxalic acid extended material Varoxan yet as a data sheet in there. The newest edition will have that. The Varoxan is a brand new product. It's using oxalic acid and keeping it in the colony. That's quite effective now in April, as is drone brood removal, splitting colonies, certainly, brood breaks, another method, or using one of our other pesticides is also another method. But the Varoxan, as oxalic acid extended, I think will have a real important addition to the way that we help keep those mite numbers down and do our spring mite control. All righty, those are some of the things I wanted to say. I think we have some time. I have time for questions if you want to stay. I think I've about used the hour up, but I'd be glad to stay and answer any questions. Bree, have you had some questions that have come in or no?
Yes, we do. We do have some
Oh, righty.
questions. So the
Okay.
first one is, could you discuss your feelings about using powdered sugar as a natural way for treating for Varroa?
Very ineffective. You need to powder sugar and continually powder sugaring. Any powder sugar that gets into the open brood, the larva particularly, is bad for them. You'll lose some larva. It is like the screen bottom boards. It's a very low level mite control. By itself, it's not going to be adequate as if you're using it singly. The only thing you're using for mite control.
Okay, great. I have another one here. During cold temperatures below 0 to single digits Fahrenheit, with the choice of opening the hive to resupply resources like Hive Alive, what would you recommend?
Very quick opening colonies. Certainly, it should be around 50, but to put that fondant shaker in of Hive Alive, you can remove those covers very quickly. Not taking frames out, not exposing the bees, not moving frames around, certainly, but uh, feeding the fondant sugar, Hive Alive, feeding a dry sugar, for example, or what number of our people in Oregon do is they feed a sugar cube. They make a great big lollipop, basically, of just dry sugar with a little bit of moisture, a little bit of water, and put it in a rim at the top, or put it in pie plates and put 2 or 3 of these pie plate sugar cubes on top to replace those if they've eaten them up. So just really top quickly, you can get an idea of how many bees are there. If you use a sticky board underneath a screen bottom board, you can go ahead and remove the sticky board, take a look, see how the bees are doing, how big the cluster might be. You'll see if there's hive beetles, you'll see if there's wax moth, you'll see if there's mice in the colony, all of those things as well.
Okay. When one has a deadout from PMS or parasitic mite syndrome, do you need to scrape those frames out or can you reuse them as is?
Good question. Thank you. I should have mentioned that. So the Varroa need to be on a live host. So if all the bees are dead, none of the Varroa are going to survive. Most of the viruses, the DWV viruses certainly do not survive outside of hosts. Their viruses are the ultimate cell parasite. And so again, if all the bees are dead, there's not going to be any virus that remains on the comb. So what we recommend is if there still is bees in the cluster, to brush those bees off, particularly if they get moldy, just brush them off with a brush or shaking them off. And then feed the frames to colonies that are a little bit stronger. If you have like a PMS dead out of a colony that was just booming in August, just great colony, produced lots of honey, and all of a sudden the mites have caught up with them and they go down fast. You're going to have several of those frames that look that ugly frame that I showed in a couple of slides. Don't give all of those frames to a brand new nuke or to a package or to a weak colony. Feed those frames in. The dead brood that's in the colony, the moldy bee bread that's in the frames, in the comb itself, the bees will clean that out. Just let the colony get a little bit stronger, and then put in a couple of those frames at a time. Let the bees clean them up. Some people want to go ahead and use like a Clorox solution, a vinegar solution to wash the combs or dip the combs in. That's probably not giving you any extra protection, but if you feel like you're doing something and it makes you feel better, it's probably not doing any harm. You just let the frames dry before you put them back into a colony. But we're not at a high enough bleach or a high enough vinegar solution to really kill anything that might still be there. So it's it's a lot of extra time and waste of energy that really isn't gaining you much more. But again, if you want to, there's no harm generally in doing it.
Under what conditions would you not reuse drawn frames from a deadout?
If there's a really dark comb that's been in colonies, gone through several years, 3, 4, 5 years of brood rearing, very dark, you could go ahead and cull some of those frames. Culling means you're going to take the wax out. Unfortunately, there's not very much recoverable beeswax comb in those combs. You can, you might not want to use it for your finest candles or your best beeswax cream, but you can recover some of that. So it would be going to your culling schedule. Uh, I mean, even if it's pretty dark, if there's a fair amount of honey in there, that honey can be, used by the bees. Even if the honey is appearing to be crystallized in the cells, the bees can re-liquefy it. So it's mainly dark, older brood comb, I would call. The rest can be recycled, fed back to the bees, let the bees clean it back up, polish the cells, get it ready for brood rearing again.
Why are there perforations on the brood cells?
Another great question, and I'm not sure I have a good answer. What we think the bees are doing, they're looking for resources. They partially remove the capping, determine that there's no queen inside. One bee does that, and then she goes on somewhere else, and then they don't pull the whole capping off. But those perforated cappings are very, very common in that situation where the colony has died and there's brood left behind.
Can mice spread pathogens to the bees? Other than cleaning out the nest and taking measures to keep the mice from entering the hive, what other measures should be taken? Does the hive need to be scorched or otherwise disinfected?
Um, no, as far as we know, the mice are not spreading any pathogen that will be a disease condition for the bees. The mice will make a mess and they will then make a territory on other combs, and the bees are much slower to clean that up. So that may be more effective to try to disinfect with a Clorox solution after you take it out. And of course, clean the nest itself out. One of the unfortunate things that happen if in the mouse in her nest choose out some comb, what the bees will do often with that is they will replace that hole that the mouse made in combs with drone-sized cells. So now you have a comb that might have been drawn beautifully and had mostly just worker cells, now with a patch of drone-sized cells. And so, Annie, I guess I should amend my question in terms of my answer in terms of the culling. I would also consider in culling combs that are older and darker, that also have more drone-sized cells, particularly, if a quarter of the cells are drone-sized cells, that's a good comb to cull out and not give back to the bees.
Do you have a photo that shows what mite poop looks like in the bottom of a cell?
It does. It is oval. I don't have one I can pull up right away. It is smaller than rat poop, certainly, but that will show up as these sort of a cylindrical, dark masses. It's bigger than bee poop. It's certainly not pieces of pollen, certainly not body parts of bees. It's a bee about the equivalent of about the second or the smallest pair of wings. So it's not inconsequential in size, but it's sort of an oval, dark looking sort of if you have a hand magnifier, sort of looks like it has a structure to it, like a pimply structure on the surface. It's not a polished surface. Sorry, I don't have one I can pull up right away.
When you're talking about pesticides, are you referring to direct spray or pesticides in nectar and pollen?
Yes, both. Certainly, colonies that are sprayed directly, whether intentionally or not, by drift of the pesticides, that's not good for our colonies. But what we're talking about with that stressor from pesticides is the amount of different pesticides that bees that are foraging on nectar and pollen are exposed to, particularly agricultural crops, although other things such as roadside department spray vegetation, community spray vegetation, tree firm companies spray trees, etc. So it is both that the accumulation, but primarily the accumulation of this incidental pesticides that the bees are exposed to. In most of our agricultural crops, and here in the I-5 corridor, we've got over 200 agricultural crops. Many of those seeds are coated with a neonicotinoid, so that you can't buy, for example, corn to plant in your garden that's not already coated with a pesticide, with a neonicotinoid. Soybeans, the same thing. Beans, the same way. Squash, some of the pumpkins, for example, they're all sprayed in advance, and it's you really have to search for just plain seed or your own seed that you're keeping back, from your own growing season. All of that is neonicotinoids, and much of that is there to protect the seed in the soil and in the early growing stage. Well, many of those plants bloom fairly early in their cycle, and there's enough neonicotinoid that it gets into nectar and or the pollen. And so the bees are exposed to those continually. We're mostly talking of insecticides, but however, we do find both herbicides and fungicides when we take a look. And if you want to look at some information, look up a paper by the Cornell professor Scott McNabb and McCart. I'm sorry, Scott McCart. And you will see that he says that we have an average, our bees have an average of something like 7 different pesticides when they take a look at the bees themselves and the bee bodies. Now, some of these are at very low levels, but it is that continual exposure, that level exposure of pesticides that kill the same way that overwhelm the bee's immune system. Bees don't have an immune system that's very strong. Their immune system is their social structure, so that some bees will die from disease, some will die from foraging accidents, some will die from pesticides. But the way the bee colony survives is that there are more that are being born continually. So that's a social immunity rather than the individual bee having a strong immunity that it can die, it can avoid being killed. A few of the pesticides kill bees right there, out in the field. Boom. They hit the flower, boom, they're dead. But by and large, the vast majority that are carried back, it's mixed into the bee bread. And of course, the bee bread then is fed on by the young bees. That's what makes up their brood food, gets into the brood food. And so the bees get fed pesticides in their food as pupae or as larvae, I'm sorry, and then in their adult exposures. So there are a few real bad actors among the insecticides, but what we're generally talking about is this whole stressor from this whole mixture of different pesticides that they're exposed to in a single season and repeated seasons.
Okay, thank you. Is there a loud sound that bees will make when there's possible queen loss? This beekeeper had a late fall dead out that was louder than other hives.
Yeah, we talk about a roar of colonies when you open it up and you're inspecting the frames when they're queenless. It's subtle. It's not hugely different, but we can pick up that sound. When colonies have queen cells, there's another series of sounds that the developing queens will make, quacking or piping, queen piping. And that's a high-pitched, very distinctive sound. Colonies that are open quickly in the fall or over the winter will have a roar to them. So one of the senses that will help you become a more successful beekeeper is listening to the sounds, listening for those sounds of things such as queenlessness, of bees that are disturbed for one reason or another.
Okay, great. Can you talk about colonies that have died because of nosema infection? And what do you do with your equipment that has bee excrement all over or inside the hive?
No, I can't talk about nosema because we don't know what's going on. Our familiar nosema, which we had back when I started keeping bees, which is now many years ago, was a species called apis, and often that what we would see is bees letting go of their uh, rectum contents, in other words, pooping inside the colony. And as soon as they got airborne, they would let go of their poop. And so there would be spotting on the colony, on the cover of the colony, on your vehicle that you drove into the apiary, on houses nearby, et cetera, et cetera. However, Nosema that species has been replaced by another one, and generally it does not cause this release of their rectum contents either inside the colony or immediately as they fly away. So we don't see this spotting from bee poop on the colony, on vehicles, on houses, and that type of thing. Not with this new Nosema. The only way we can detect nosema, and most colonies will have some level of nosema, but we're looking at a level of nosema that seems to be significant, and that's better than 1 million spores per bee. And not every bee will have this. What it is, it's a type of fungus that lives in the bee gut, and it steals the bee's food before it can get out of the gut into the hemolymph of the bee. So it's living there in their gut, and it's eating. That's what it's eating on. And that's why the bee feces builds up. At least it did with nosema. So if you see dysentery, you see bee poop in a colony on top of frames, for example, on the outside of the comb itself, that would be good to go ahead and try to use that Clorox solution or the vinegar solution try to wash it off as much as you can. Now, we think most of the bee poop that you will see these days is not nosema. It is rather uh, the foods that are available, whether it's the bee bread, whether it's the nectar sources that have been ripened into honey. So that's what we think is causing the dysentery, bees that are letting go their poop inside the colony that they can't get out and get rid of it, not nosema. As I said, I can't say much on nosema because our antibiotic that we used to use, a material called Fumidil B, the antibiotic fumagillin, if you feed it to our bees, the newest nosema species actually comes back at a higher level. So you're actually doing harm by feeding the antibiotic, the fumagillin, to a colony. Um, the good news is we find it in certainly less than 5% of the colonies. This heavy losses that our commercial beekeepers have had the last 2 seasons, they have samples of bees, of dead bees, of dying bees, and they will make an analysis of nosema to try to determine if nosema was or possibly could have been one of the factors for this heavy loss. Going back to the CCD losses that we had in 2006, 2007, nosema was thought to be one of the factors, along with virus disease from bees, was thought to be the factor that might have been the reason for the heavy losses back uh, starting 2004, 5, 6, 7, 8. We don't see too much of the CCD, but we're seeing the PMS these days.
While on the topic about nosema, we have a couple more questions. Do you recommend routine nosema medications?
No, do not, because of the factor that the condition comes back worse after the antibiotic has worn off. No, I do not. No, we do not.
And what do you think about the prevalence of Nosema ceranae in spring?
We know it's in some colonies. We know it's in some bees. It's at generally at levels that are below that one, on average, one per, 1 million spores per bee. Although if you look at 20 bees, you squash 20 bees, maybe you'll find one or two that if you squash the bees, whole bee bodies, sometimes bees with nosema, their digestive tracts will come out and be like scattered. It kind of looks different from the rest of the bees. So 19 bees look like squash bees, and then one will look like this squashed and scattered remains. That one generally would have the nosema. But if it's only 1 in 20 bees, and depending how high the number is, the average for those 20 bees is below that 1 per million. So we can find nosema in most colonies if you squash enough bees and look at them individually. But colony-wise, we're finding certainly less than 5% that have any level of nosema that might be of concern.
Okay, and then there's a couple of questions about dry diarrhea on frames. So, if you're seeing diarrhea on frames in a deadout, what can be learned from that, and can you use those frames for other colonies?
Uh, yes, generally you sometimes we'll see that diarrhea, that the bee poop on the colonies that have too few bees, those really small clusters of bees. Often we'll see it right there on the top bar, right where that cluster is, those remaining, moldy bees. It should be removed so that if you have a wooden frame, I would actually use the hive tool and scrape it like with the curved edge of your hive tool. Scrape it, scrape the sides of the frame if you have wooden frames, and then go ahead and wash it with a bleach solution, using about 2 to 3 tablespoons per gallon of water, so a fairly strong bleach. But mostly we'll see it with those clusters that had too few bees. Can you give them back to the bees to clean them up? The short answer is yes, but I would not want to have a weaker colony have to clean that up. So it'd have to be a stronger colony. And only like in the summer when they can get out and get rid of any waste that they accumulate from the cleaning bees that accumulate any waste.
We have a few people asking about recommendations for Varroa treatments in spring, and one person added that they're worried about formic because of negative effects with queens. What
Yeah,
else would you
it's tough.
suggest?
Yeah, formic is tough on bees. It's tough on open brood. Um, if you use one pad for the first 10, 14 days, and then use the second pad for the by itself for the remainder of the time for roughly 21 days, it's less likely to cause problems with the queen and open brood. Not necessarily no risk, but less. If you use the two pads, particularly in spring, often you'll find that the queen will stop egg laying for a week or so, or will kill some queens and it will kill all your open brood. And of course, that's kind of critical in spring. We want the colonies to grow. So formic of all of your choices in the spring is the toughest one to try to use. Um, but if you're going to use it, we do, and it's permitted on the label, we do recommend the one pad the first 10, 14 days, followed by the second pad to try to avoid that collateral damage to the queen and the brood.
Are all paralysis viruses spread by Varroa?
No, not all of them. Uh, we know the DWV viruses are, the Israeli is, but it's an open question on the chronic and acute. And then there's there's, bees have over 20 viruses, and so the Varroa generally does not spread most of the others, as far as we know. Any researcher that's looking at viruses will use as their control a virus called Lake Sinai virus, because that is spread by varroa mites, but it doesn't seem to be pathogenic to the bees. It doesn't seem to harm bees. So when they're doing experiments, that's one way to try to do as a control, to infect them with a one virus, and then your second virus, this other one, used as your control. So no, not all, but yes, all the variants of the DWV viruses we think are spread, or they're spread enhanced by varroa feeding on the bees.
Do you suggest testing for mites or just treating for them?
I suggest testing, and then on the basis of the testing, make a decision in terms of what you might want to use as a control, or give you peace of mind that your mite levels are at such a point that you don't need to control. Now, I have to add a rejoinder to that. If we test in this month, like tomorrow, the next couple of days when it's nice weather, if we test in April, if we test in May, and if we test in June, generally we're going to find very low mite numbers. Now, our test is generally taking a sample of 300 adult bees from the brood area, then washing them with alcohol or non-sudsing soap, or using powdered sugar to wash the mites off the bee bodies. But early spring, our numbers are zeros, our ones, our twos. Unfortunately, that is an underestimation of the reproducing population. Next month, in April, most of the mites will be either in drone brood, or a limited amount will be in worker brood. And the vast majority of any mites, adult mites in the colony, 75% or more, will be in a capped cell. So if you wash 300 adult worker bees, you are missing all the mites that are on the drones. You are missing all that 75% of the mites that are in the cap cells. You're only going to be able to look at roughly 25% of the mites. And as the colony grows from 10,000 bees. In the end of April, that's going to be 20,000. The end of May, that's going to be closer to 40,000. And you're still taking only 300 bees. So it's getting harder and harder to find those mites. So sampling during a winter, it's risky to open a colony and take out a sample of 300 bees. And in early spring, it's underestimating the level of mites reproducing in your colony is not a very effective means of determining how many colonies your mites have. Now that changes in July, August, September. So sampling 300 adult bees at that point, worker bees, will give you a decent measure of what's going on in the colony. You'll usually see the recommendation to sample every month. Uh, if you have a very large mite population, that number can jump up drastically in a 2 week period of time. So that one sample in July, one sample in September, maybe one more sample generally is not enough. So we recommend at least monthly sampling July through October. In some cases, for some colonies, preferably maybe twice a month to really know what's happening to the mites in the colony.
Okay, and will you find mites on bees if you do an alcohol wash on a deadout? What happens to the mites in a deadout?
No, they don't separate. They don't separate. So no, we don't do the mite wash on a dead out. No, many of those
Okay.
mites will fall to the bottom board and it'll be in all that mess of bees on the bottom board.
Okay, and then going back to the Varroa treatments in spring, this beekeeper doesn't like using Formic Pro or Amitraz, so do you have any suggestions for another treatment appropriate for spring?
The time oil products, Apagard, Apalife R. In my survey, I asked for maintenance. People that use Apalife R, particularly in the spring, end up with the lowest levels of colony losses year after year after year. Apalife var is thyme oil and some other essential oils as well. You have to go in the colony 3 times because it's only a week-long effectiveness of the wafer that you put into the colony. And they're both fumigants, so you put them at the top of the colony. The Apagard is a gel, and when the first glop of the gel is gone, you put another glop in, or you put another tray in. You can buy individual trays. So that's a pretty good effective treatment. The other one that I think that everyone should really strongly consider using is the Viroxan. Viroxan is the extended oxalic release material. You put in these fiberboard hanging pieces that look like cardboard. You hang them over the top of the frame. You put one in for every 2 frames of brood. So you may start with only 2 or 3, but then you increase the number. Generally put them in as early as April, switch them out in May, and then often many people will switch them out again in June as they're putting supers on. And that keeps a low level of oxalic acid constantly in a colony. Now, if you are a mind to not try to put as much acid into your colony and not use a pesticide such as oxalic acid, drone brood removal. Drone brood removal is also an effective way to reduce the mite population early in the spring, reduce the mite growth of the reproducing mites. The mite founders mites really, really select the drone brood first thing in the season. So that if you are roguing out drone brood once it's capped, then you are really keeping down the amount of reproduction of those mites that survive over the winter. So if you do this in April as they start rearing drones, continue it into May, uh, uh, you can find the drone brood in your colony and take a capping scratcher and just impale the drones and pull them out through the cells. uncap the cells, and the bees will then yank out the developing drones. And of course, that interrupts the mite reproduction. Or you can use a whole frame, or what some beekeepers will do is use a medium frame and a full depth box. The bees will build comb underneath the bottom bar, usually drone comb. If the queen comes there and lays brood, then you can remove that drone comb once it's in the cap stage. Mites do not enter the cells until just before the cap stage, so that you only want to be removing capped drones from their colonies. And then finally, uh, letting your colony swarm, or you doing that same operation, and that is doing something like making a colony split, and that is what we call a brood break. What you can do is in your brood break is put most of the cap brood in one of the splits that you make, and leave most of the open brood in the other split that you make. And then in the one with the open brood, you can go ahead and do again oxalic acid, because that will remove many of the adults. And if there's no cap brood, it can be quite an effective treatment. And then leave the queen in the split that has the open brood, so that the cap brood in the other split will then emerge from their cells. And at some point, there is no cap brood there, even if you add a brand new queen, or let them raise a queen themselves, or put all the cap queen cells in that split. And there'll be a period when there is no cap brood, and you can again use oxalic acid. And oxalic acid is the one that we use during the winter period when there is little brood or no brood. And it's a very effective tool in that way. The extended oxalic is an effective tool to keep a low mite number low. So it's really interfering with the reproduction of mites. But if you have a high mite number, oxalic and brood, oxalic is not the best tool to use. You have to use it repeatedly. You have to use it every 4 or 5 days for about 6, 7, 8 times. So for the next 4 weeks, you use oxalic acid, 7, 8 times in your colony. That's a lot of acidification of a colony. And the high
Okay.
acids, whether using formic or oxalic, the apivar will not work very well. In other words, the amitraz, that's the apivar plastic strips. If you've used a lot of acid in your colony, formic or oxalic, that apivar, that amitraz, is not an effective tool. The acidification somehow affects it, and is not as a good killer of mites.
Oh, interesting. I didn't know that. Should frames from a deadout be scraped clean to remove dead brood, moldy honey, etc.?
No, just brush off the adult bees on the outside, those moldy adult bees, and then let the bees clean out the cells themselves.
Are large numbers of dead bees piled directly outside of a hive a sign of a particular cause of a deadout in fall?
So dead bees piled at the colony entrance could signify pesticide loss. Generally, bees in fall, when there is forage that have had mites feeding on them, they leave the colony and don't pile up right at the entrance. Um, so generally, no. It is an indication of robbing, but it could be pesticide if you're in an agricultural area, but more likely to signify robbing, another colony robbing from the colony where you see all the dead bees out front.
Okay. So we just have a handful more questions. Does insulation at the bottom matter? I've heard from several beekeepers that they keep the bottom of a screened board open year round. What are your thoughts?
So the screen bottom board is a sort of a passive way to control mites. Mites that are still alive that fall through the screen, then can't get back on an adult bee. Not really effective. It gives you 3 to 4% better winter survival than if you don't use it. However, in my surveys of the last 15 years, I've been asking the question, so do you close the screen board or do you leave it open? And the people that say they close the screen board, some only in the coldest days, but do some measures of closing that screen at the bottom, say that they do have better winter survival. And it's another 3 to 4% boom. So if you have replaced your solid bottom boards with screen bottom boards, and if there's any possibility that your colony is open at the bottom, in other words, cold air can circulate below that screen bottom board, it does seem to make a difference if you, at least during the coldest part of the winter, try to close it off. You don't have to, put it solidly, but sometimes just using the mite debris board is enough to do it. But it would indicate that, yes, probably closing a screen, not leaving the screen open year round, would give you a bit more better chance of winter survival. The survey says.
Do you recommend using sugar cubes or 1-to-1 sugar water in winter? Will the sugar water have too much moisture?
Yeah, that's the problem. The sugar water during the winter is high moisture. We think moisture is an issue, particularly in the I-5 corridor. Maybe not over into the drier parts of the state, certainly, but certainly the corridor, the coastal areas certainly as well. And so the sugar cube is a good alternative. It's an emergency feed. It's peace of mind. What will happen is that that warm air from the bees leaves the bees, goes upward, and then hits that sugar cube, and will make a slurry out of that cube on the underneath side of it. And so then the bees can take their mouth parts, which they can't chew with their mouth parts, but they lap. So when that gets to be that slurry sugar underneath the cube, that's what they lap up with their mouth parts. And whether you use fondant sugar or whether you use plain, cane or beet sugar, doesn't make a difference. Many people do like the fondant sugar. It costs more. Um, or you get the real convenient Hive Alive. You buy that product and feed those over the winter. It's a good emergency, a good peace of mind that just in case they don't have enough stored honey or it's way over here in this frame, that they can't get to. It's a good insurance policy.
Okay, we have one person wanting to know what your shirt says.
It says, I survived. I survived. Okay, including COVID-19 and murder hornets. This was from our meeting that we had out in Othello. This is the WASBA meeting. Okay, the first virtual conference.
Awesome.
So I'm
Uh,
appropriately attired for was Washington beekeepers tonight.
could you talk about tracheal mites briefly?
Tracheal mites arrived in the U.S. prior to the varroa mite. Um, some of the regulatory activities tried to kill colonies, and they were trying to kill colonies, and a number of colonies did not do well initially with tracheal mites, but we didn't really have an effective control. And so something like menthol, we would make a menthol cube and put on top of the colonies, um, but not really effective control. And so what happened, we believe, with tracheal mites was after their arrival, um, because we didn't artificially try to control, some beekeepers lost a number of colonies that were susceptible to tracheal mites from the queen breeders and the package and nuke suppliers. They died. And then the bees acclimated to tracheal mites, and it became a non-issue in terms of being very serious in terms of continuing to kill colonies after those first few years. The varroa mite story is very different. Within 4 months, a chemical company had the development of amitraz. And so when varroa mites came in, beekeepers didn't want the beekeeping authorities to kill their colonies. So the beekeepers immediately started using chemical control, and that was called apistan, fluvalinate, and that was actually a pretty good miticide until the bees developed resistance. But the story of the two mites is just completely different. The bees are taking care of tracheal mites, and as beekeepers, we've sought to take care of varroa mites, and we're doing a lousy job. Varroa
Does
mites
using a
came in '87, and we're talking how many years have we been talking? 35 years of this method of controlling, that method of controlling, preferred method of controlling, too many pesticides, not enough inside of the county, resistance of the mites. 35 years with this one pest.
does using a smoker on a hive or colony help pest, parasite, disease, or virus avoid detection?
No, not to our knowledge. No, there's nothing you can put in a smoker that will be of a concentration high enough to affect any pathogen or pest control.
How can you raise drones for queen breeding without getting super high levels of varroa?
You can't, and that's basically our issue of open mating. You really can't. So the what we have to do is control mites well enough on the drone population that we have enough drones available for mating. Um, it's tough because when we want to raise the drones for mating is when the mites want to go into the drone cells to do their reproduction.
Okay, and then the last question we have here is someone in Colorado is wondering when they should start testing for mites in a year. Is it like when they first get into the colonies in the year?
Um, so when you first get into colonies in the year, whatever that is, April for where you might be higher elevations, maybe, not to May, the colony population is not very strong. Most of the mites are reproducing in drones or are on the drone adults, so that sampling early in the year is not very effective to give us a measure of what's going on in our colony. In August and September, yes, sampling it can be a good way to make a decision what kind of control I use, or maybe I don't do any control at all. But early in the season, no. So early in the season, we're really trying to prophylactically control the mites, just as we are during the winter when we use oxalic. We don't sample the bees in winter and then decide if we're going to use oxalic. We're going to use it or not use it. So in the spring, you're going to use your controls or not use them. But sampling adult bees is not going to give you a good measure of what's going on.
So that person's following up with, so it wouldn't really be appropriate to test for mites until like August or fall.
Correct. But would be very appropriate to try to control mite reproduction as early as April.
Okay, and then we did get one more question. If or when Tropicalaps arrives in the US, will they bring new viruses or do they share viruses that beekeepers see with Varroa?
I'm not a trophy expert, and I don't know what their story is with their spreading the same viruses or if they have a different group of viruses. So sorry, there's an excellent, just a superb site on the Tropicalaps mite. If you go on to Project Apis M. If you use your search engine, Project Apis M, or go to the Apiary Inspectors of America, just use your search engine. There's excellent, excellent information on the trophy mite, and it might well answer that question. I'm sorry, I cannot.
Okay, Dewey, there's no more questions in the box. Thank you for your presentation today.
Thank you,
Um,
Bree.
and thank you all for attending. Before you close your browser, you'll be prompted for that short 5-question education and outreach survey. I would really appreciate any responses if you can participate in that. And as a reminder, we have 5 more webinars planned this year. So for more information and to register, you can visit our website. Thank you so much.
Bye-bye. Thank you. Have a good spring. And our survey.